Hey guys, welcome to Science Labs. My name's Scott and here we go.
Scott!
The science guy.
Scott!
The science guy.
Scott! 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 Scott!
The science guy.
Science rules.
Built by the science guy.
Inertia is a property of matter.
Bill Nye, the science guy.
Scott! 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 Scott!
The science guy.
Welcome back, viewers. Thank you for being here for my first ever video. Today we're going to talk about and take a look at transcription, but more importantly, post-transcriptional modifications. Transcription is a process which takes place inside the nucleus of cell. It takes place in 3 steps. First, initiation. Second, elongation. And third, termination. There's a lot more detail to these steps, but more importantly, the major thing we want to look at today are the modifications after this process. Scientist Haley, what do you have to say about these modifications?
Pre-mRNA molecules go through 3 main modifications: 5' processing, 3' processing, and RNA splicing. Let's start with 5' processing, which is done in order to add a modified guanine, which acts as a cap on the 5' end of RNA. 5' processing is done by removing the 5' phosphate with an aid of an enzyme. Another enzyme then catalyzes the reaction to produce the diphosphate 5' end. This end attacks the phosphate on the GTP molecule to add guanine and create the 5' to 5' triphosphate link, which eventually results in the cap. The cap protects the 5' end of the primary RNA from degradation in the nucleus and assists in export to the cytosol. Now back to you, Scott and I.
There are other types of modifications done to the mRNA also. mRNA is made up of 2 subunits, introns and exons. The exons stay in the nucleus and are expressed, while the introns are spliced out. They're spliced by an enzyme called spliceosome. What does this look like at the molecular level, you ask? Let's take a look. The spliceosome binds to the mRNA complex. It slowly cleaves the introns and attaches loose ends of the structure. This is useful because the pre-mRNA must become mRNA in the final of the process. Scientist Luke has some other insights about the other end, the 3' end.
Thanks, Scott and I. The pre-mRNA processing at the 3' end of the RNA molecule involves cleavage of its 3' end. This allows for the addition of adenine to form a poly-A tail. The cleavage occurs when a polyadenylation signal sequence AAUAA, which is normally located near the 3' end of the pre-mRNA molecule. The site of cleavage normally follows this polyadenylation signal sequence with CA. After the synthesis of the sequence elements, several multi-subunit proteins, CBSF, CF1, and CSTF occurs from RNA polymerase II. The 3 factors bind to the sequence elements. This complex cleaves the RNA between the polyadenylation sequence and the GU sequence. At the cleavage site marked by the CA sequence, poly-A polymerase then adds around 200 adenine units to the 3' end of the RNA molecule using ATP. As a poly-A tail is synthesized, it binds multiple copies of the poly-A binding protein. This protein protects the 3' end from ribonucleus digestion.
Can you believe that all of this takes place in the nucleus of eukaryotic cells? Find out more next week and join us at Science Labs. And remember, science rules.